I want to start by thanking the organizers for inviting me to speak. My name is Jenny Ekholm, and I'm going to talk about full-length RNA sequencing for Alzheimer's disease. Now, I want to spend a few moments first on Alzheimer's disease in itself. So, as most of you know, is the most common form of neurodegenerative dementia that is caused by progressive cortical atrophy. And the symptoms range from progressive loss of memory and deficit in thinking, problem solving, and language. Now, if we look at the genetic makeup of Alzheimer's disease, it's been concluded it's definitely genetic, as we see a family aggregation of the disease within families. Genetically, Alzheimer's disease can be divided into two different subgroups. You have early onset Alzheimer's disease, where patient starts displaying symptoms in the early 20s, and 4 genes can really account for 5 to 10% of early onset Alzheimer's disease. Whereas for the late onset Alzheimer's, it manifests later in life, and here we see a more that this type of disorder is more multifactorial in nature. Now, looking at the Alzheimer's disease, candidate genes that have been found for Alzheimer's disease over the past decades. As for many of common complex disorders, there's quite a few candidate genes, so over 20 that's been accounted for through either linkage studies or GWAS studies. And now there's several different studies that have been applied for following up on these findings. So they range from DNA sequencing, transcriptome sequencing, proteome studies, and metallome studies. And here I'm going to talk about how we actually applied a long-read sequencing to look at some of these findings. So just a few words on long-read sequencing. So there's the key characteristics, of course, is the long reads. So you're going to find that half of your data have reads that are longer than 190 kilobases, and some are even longer than 300 kilobases. Because there is no systematic errors, you also get very high accuracy to consensus to building consensus to coverage. And it also has single molecule resolution, which is important to remember for later in the presentation. And because it doesn't have any sequence context bias, you see uniform coverage across any percentage of GC. As long as you use native DNA, you can also simultaneously detect base modification. Now, how does this work? Well, you start out with a double-stranded DNA. Then you ligate sequencing SMRT adapter to each end, where the sequencing polymerase can bind. The polymerase has strand displacing capabilities, so it will open up this SMRT structure into a circle, and it will continue sequencing around this in a rolling circle sequencing mode. The benefit of this is that you sequence both the reverse and forward strand in one go, and any of the random errors that come off along the way can then be washed out through building coverage, as we talked about earlier. And this will then result in very highly accurate HiFi reads. So this technology was then applied to different ways of looking at the Alzheimer's disease. So in one study, we used targeted enrichment. So we used probe-based capture to target 35 Alzheimer candidate genes that have been highlighted through linkage GWAS studies. The great thing about using probe-based capture is that you can use the same probes to capture both the genomic DNA as well as RNA. So here you can see we have 2 patients, we capture both the DNA and the RNA here from the temporal lobe in the brain. Now, we detect in the 35 candidate genes, we detected a broad range of variants. So in addition to just regular SNPs, we identified 31 unique structure variants that range from 65 base pairs to several kilobases in size. And in terms of the isoforms, we captured over 500 isoforms for each of the patients. And here you can see an overlay. So here's the gene code, isoforms that were found in gene code at the time, and also all the isoforms for patient 1 and patient 2. And as you can see, there is not a lot of overlay between the different groups. Now, looking in more detail at the results, so MAPT gene is one of the candidate genes. So here's patient 1. And what we found is, again, because we have long reads and you can see all the different variants, including the SNPs here. that will actually help you to phase the reads, the sequencing reads into two different alleles. So now you see allele 1 for patients and allele 2 for the same patient. So what we found here is in the MAPT gene, we found a heterozygous deletion that was only present in allele 1 and not allele 2. And allele 1 resulted in 25 different isoforms, whereas allele 2 only resulted in 5. However, we did find in 3 of the 5 isoforms had a novel exon. Looking at another gene, same here for patient 1, we found a heterozygous deletion of 750 base pairs. And then looking deeper into the next level of information, which is the isoform information, we can find both retained introns and novel exon for these patients. Now, looking at the Alzheimer's disease in a different way, so instead of looking at it in targeted fashion, we wanted to look at the whole transcriptome. So we used 300 nanograms of total RNA from an Alzheimer's brain sample and made a sequencing library, then sequenced it on one wide NA smart cell that will allow you to fully characterize a human transcriptome, and then of course analyze the data as well. Now, just to iterate why would use long-read sequencing instead of traditional RNA sequencing. So as you know, on average, transcript size, gene transcript size is 2 kb. And also on average, each gene has about 8 different isoforms. This means that if you have reads that are 150 or 300 base pair in length, it is very difficult to actually know which read comes from which isoforms since they're all so similar to one another. So this makes it really impossible to assemble the different isoforms. So while traditional RNA sequencing is great in capturing gene level expression, you do not know which isoform is contributing to that expression. Whereas with long-read sequencing, you capture the entire transcript, so there's no assembly required, and you know you have captured it because you capture the 5' end, the poly A tail, and the 3' UTR. So going back to the Alzheimer's brain sample, so what we found is altogether over 7,000 different genes were identified. 96% of these were known genes, and over 3% were novel. And this translated into over 160,000 different transcripts. The transcript sizes range from 80 base pairs to over 14 kb. The mean size was 3.3 kb. But again, here you see that most of the transcripts do fall within the 2 kb range. Out of these 162,000 transcripts, only 32% were known, and 68% of them were novel, showing again the power of long-read sequencing in detecting actual isoforms, and also the complexity of a brain sample. Just looking in a little bit more detail in this data, so here we have SEN2, which is one of the candidate genes. So in red, you see the sequencing data from this experiment, and in blue you see the transcripts that have been uploaded to GenCode. So you can see, first of all, there is a much in-depth isoform diversity within this sample compared to the reference. And if you look a little closer, you can also see there is alternative start sites. We have novel exons present. We have variable UTR lengths. And we also interestingly see a transcriptional read-through. So, so then you have both the SEN2 and this adjacently located gene COQ8A that seem to be have been combined. So we of course haven't done any downstream analysis, but the next question would probably be, do these co-express as well? So then looking at the APP gene, which is another the candidate gene. So here again, you see quite broad transcript diversity compared to the reference, but we also see exon skipping present, which we also see some evidence for in the GenCode database. And just looking at a couple other genes, candidate genes, so we have SOL1 and ABCA7. Again, you see a much broader isoform diversity for both genes, and there's several new alternative splicing taking place and complexity, alternative isoformic complexity taking place when looking at this with long-read sequencing data. So we wanted to also put an additional layer of complexity, look at an additional layer of complexity for the Alzheimer's disease. So we used the long-read sequencing to generate full-length transcripts, but looking also at single cell level data. So the great thing about single cell sequencing is that not only can you see now which transcript is present in the genes, but you can also see which transcript is being expressed at each of the cells. So you're adding a additional layer of complexity. And just to kind of show that there's a great concordance between Illumina and smart sequencing data, we did a side-by-side comparison, looking at the barcodes being picked up, and we found 100% concordance. So just to quickly walk through the workflow for Iso-Seq single-cell sequencing. So first you start out with capturing the actual single-cell cDNA. Now this can be, any single-cell platform can be used for this part as long as they can capture full-length cDNA. Once you have the ready PCR product that consists of 5' end, transcript, poly A tail, unique molecular identifier or the UMI, and then a barcode to distinguish which cell, which transcript comes from which cell, and the 3' end. Then you're ready to proceed into smart sequencing library prep, where you just add the sequencing SMRT adapters, after which you do sequencing. So unfortunately, due to the COVID-19, we were not able to fully finish this results. So these experiments are still ongoing, but I'm going to show you what I have so far. So again, this is kind of comparing, showing some of the results just on a high level. So again, here we have the bulk RNA that we just talked about from the Alzheimer's brain. And here we have a single cell experiments, one on the human brain organoid and one on the human cell line. And just comparing these, what you can see is, because you have the transcript length here on the x-axis, you can see that there's definitely a shift towards the shorter ones. So this is something that we can see, that depending on which single cell platform you use to sort your cells, there is definitely preference towards the shorter fragments. So just to show some of the results that we found, so here's just a quick look at the TROPOELASTIN gene. So this gene is known to only be expressed in astrocytes, and that is exactly what we found. So we only found isoforms for the astrocytes and none of the other cells. And if we look at the complexity of the transcripts found, we can see that a lot of them are present that we've already seen evidence from in the gen code. But again, the great thing here is now you know which cell adds to which transcript complexity across the data. So while we are working on finishing the experiments, I also wanted to draw your attention to one of the publications that use the same method to look at single cell data. And here they actually did highlight one of the Alzheimer candidate genes, the BIN1, showing again the same thing, that using single cell data, it's very powerful to pinpoint what cells are expressing what isoform. So for the orange exons here in BIN1 gene, you can see that they're mostly just expressed by neurons. Very few of them are expressed by astrocyte, microglia, oligodendrocyte precursors. So to conclude, RNA long-read sequencing offers many benefits over traditional RNA sequencing. With Iso-Seq, you will be able to capture the entire transcript, starting from the 5' end onto the poly A tail and finally to the 3' UTR. Therefore, there is no need for isoform assembly. In addition, using HiFi reads, the data is highly accurate, and this will allow you to identify variants, which will also help you to phase the two alleles. So all in all, you get much more complete view of your data. Iso-Seq full-length transcript sequencing can be used in many different ways. You can use it in a targeted fashion, either by applying a reverse transcriptase PCR, or as I showed earlier, probe-based enrichments. Or alternatively, you could also choose to look at the whole transcriptome, which you can now do in 1.8M SMRT cell. Or you could even choose to drill down one level further and do the whole transcriptome on a single cell level. And this would then add more biology to your conclusions. With that, I want to thank you for your attention. And if you would like to learn more about RNA sequencing using long reads, please visit pacb.com/Iso-Seq.